principle, the YouTube broadcasting will start now. So let me check that. Yeah. So perfect. It's more or less 5 minutes past 10, so I will start. If anyone has any question, please ask or stop me, okay? So thanks for being here. I hope everyone knows why I choose the subject of viruses for today's training biology. First of all, I will start with a question. Did any one of you know which are these two viruses on the screen? I mean, one should be more or less obvious, but the other one, I don't know. The left one is one type of corona. That's the main thing. I'm saying this because there is a lot of let's say not the correct use of the words. This is the SARS-CoV-2. A lot of people think that is the COVID-19. COVID-19 is the disease, and this is the name of the virus. So I know that the virus's naming is strange because they are not considered living things. So they are not using the typical family and genera naming. And the right one is a phage, yes. In this case, it's a lambda, but it can be also the T7. But I think T7 is shorter. So what is a phage is a virus that affects bacteria in this case, okay? So we don't have to be worried about this one, at least not for our health. Sure. I more or less make this schedule. I think I changed it in the last minute because while I was doing all of this, I see that the CRISPR part is longer than I was expecting. But, well, I think it's okay. And then the Phage Display, the last part, it will be shorter, not because it is, but I think it's easier to understand. At least for you it will be, I think. So I think at the end the break will not be in the middle of the presentation, more in the last part, let's say, but I think it's fine. Well, of course, we need to start about what is a virus. If at any point I said that the virus are, I don't know, organisms, it's my fault, because viruses are not considered living things or living organisms. So my bad, and if it's in the presentation, it's an error. Okay. So because viruses are obligate infectious particles, let's say, or parasites better, because the particle it's called virion. We will see later. It consists of a protein code and the genetic information inside. That's all. They can be more things, but this is the basic parts of the viruses. They can be RNA or DNA inside. So maybe we have a virus that it's only RNA, and in order to be infectious, they need to be converted to DNA first. We will see about that. So what we need is an origin of replication. So imagine a plasmid that we use for infect the bacteria in the lab. So this may be a typical example of a DNA with an origin of replication. And then the protein code, that it's mainly for the protection of the DNA, but also is important for the infectious mechanism. Yeah, and every genera, every organism has some virus in the environment. So also bacteria can be infected with virus. And this is one example. This is one of the less sophisticated virus. This is a filamentous virus, sorry, and it consists of more or less 10 genes. So 10 genes is one of the minimum number of genes that it's inside viruses, and that's all. These genes can be important for the replication structure or the assembly in general, so the mechanism. And they are, well, for some viruses, these are really well described. Okay. So first of all, I will talk about the capsid, that is the protein outer part that brings protection to the DNA or RNA. And normally they are made of what we call capsomers, so they are identical proteins that can assemble in a shape in which the genetic data can be inside. Okay, and we have a lot of morphologies. Viruses, as you said in the first slide, the introduction, we have different morphologies depending on the virus. So we have the helical or I cannot pronounce this word properly, and not in this time at 10. So these are the typical, like the icosahedral is the SARS, for example. And then there is all the rest that we include in the category like complex. Complex means that they have membranes also outside, glycoproteins, like if we return to the first slide, these parts or protuberations here, these are the capsomers in the grape, and the red and orange are more proteins, another coat, another layer that is for protection and to avoid the immune system in the body. So this is not mandatory. So this is considered a complex virus. Also, the phage that we see is also considered a complex virus, because there is the icosahedral part or the prolate part here on top, and all the other parts are more for the infectious mechanism. So it's also considered a complex. There is also giant viruses like this, and they can have like different morphologies. This is like the pox virus. Well, the name is also on the image. And these are, like I said first, the easier or the more simpler viruses that are the helicals or the filamental viruses. Is the same in this case. And it consists basically in one protein that coats the whole DNA, and then there are some 2 or 3 more proteins for the infectious mechanism. So it's really simple. One example of this kind of virus that affects humans is, for example, the Ebola virus. So the simplicity is do not refers always to, well, this affects only bacteria or this, because are much simpler. No, we have an example like the Ebola. Okay, all the virus has a cycle. We have mainly 2 cycles: virus cycle, or it's not life cycle because again it's not a live organism, but they have a cycle that we call lytic cycle or lytic cycle. I will say lytic because it is easier for me, but maybe it's not the way to say it. And then there is a variant that is the lysogenic cycle. These are the stages of the lytic cycle. I will be really vague in this part because it's maybe not the most important part for us. So there is a lot of things here happening, but I will simply enumerate the parts of the cycle. Obviously, all the viruses need to attach and penetrate the cell, so inject the DNA or RNA inside the cell, and this needs to be well, to use the mechanism of the cell to generate new capsids and new virions, so new infectious particles of the virus. This is the biosynthesis part. Then they need to maturate, so the DNA needs to be inside the virus, and then they can break the cell or simply exit the host cell. This obviously cancels completely the cell normal life and stops working properly. And in the last step, the cell normally dies. So this virus, the virus that follows this lytic cycle are more virulent. So, well, that's the first. And the second is the lysogenic. The lysogenic is the same, but with a new step in the middle, that is the prophage, that is a dormant step. We have a really known example of viruses like this, like HIV. Okay, the HIV infect the cells. It has like a small lytic cycle, so this infection spread to the body, but then when the most cells that can infect are already infected, the virus starts dormant. So the DNA, the DNA in this case, gets incorporated to the cell, so the host DNA, and simply do nothing. And once in some stressful conditions, this DNA, it gets activated again and starts the lytic cycle again. This is maybe, I don't know, 20 years of dormant state. So, well, as you know, the HIV can be a lot of years after you get infected. So, well, this happens also in bacteria, and we have a lot of examples. I think the T7 bacteriophage is also a lysogenic virus, and it's used on the lab because the conditions in which you can activate the production of the virus are well known, so you can use it in advantage for the expression of the virus. So maybe it's useful on the lab. Well, okay. We have a lot of classes, and the organization of the viruses are not as straightforward as it's for the living organisms, because the viruses are always depending on the organism that they can infect. So maybe we have some ones that are really not complex and not really evolved, but they are new viruses. So it's really strange, and we cannot organize the virus in a genetic way, let's say. So we have a lot of classifications, like for the shape, for how the DNA is inside, or its RNA, or what is the cycle that they are doing. And one possible is the Baltimore classification, that is this last one, that depends on how is the genetic data stored inside the virus. We have the group 1, that is like us, let's say, a DNA and a double string DNA. So we have also a type 2, that is a single molecule of DNA. And then we have RNA viruses in a positive coiling or a negative coiling. And then the RNA viruses or DNA viruses that needs to be transcripted again, so retranscripted or reverse transcripted to be DNA again, maybe because they are lysogenic, so they need to be incorporated in our DNA. These viruses are special because they need, the reverse transcription, the protein that does this reaction is not present in normally the host cells. So these viruses need to also carry this protein in the virion part. So if this is not well done, this particle with capsid and also the genetic, but without the reverse transcriptase, this will not infect. Okay, so maybe this is not the best group to work with, but these viruses exist, and they are kind common, I will say. Okay, for us, it's more or less evident that we have mechanisms for defense ourselves from the viruses, that is the immune system. But we also saw that bacteria and archaea can also be infected with viruses. So what mechanisms they have? And the main one, or the most important one, is maybe CRISPR. CRISPR is, I think for sure you know about CRISPR, or you heard about CRISPR, you read about CRISPR in the papers, because it's a really important mechanism, and it's being developed for doing a lot of things right now. And I have to say that it's much simpler because they can be a standalone mechanism in these single cell organisms for protecting against viruses, and not only one, so a lot of them. It's like an immune system. We have some immunity to some viruses, to some bacteria, to some agents, and this CRISPR can also protect the cells from all of these, a lot of sources of infection. So the CRISPR locus, the locus is the space, the genetic space, is like this. We have these Cas genes, so the genes that code for Cas proteins. We will see some of them and what they do. Then we have a promoter or a leader in order to induce this part over here, and this is the main part, the part that can give protection to the cell. Okay, we will focus more on this part, but for you to know, it's important to know that this part is a repetitive part. So we have a repeater and a spacer. Then the same repeater, so it's the exact same code here, genetic code, and then another spacer. This is not the same spacer. We will see how these spacers are added to this DNA part. Okay, these are the proteins Cas1 and Cas2. These are the main proteins in the reaction of get these spacers on the gene. Okay. Okay, imagine that this invading DNA is a viral DNA inside the cell. So this is happening inside the bacterial cell or an archaea cell. So Cas2 binds this DNA, and Cas1 cuts the DNA, and also cuts the spacer DNA in these PAM regions, also here and here. That is a really known part on the DNA, typical part on all the DNA. And then this is attached inside like this. So one chain is on the left, or the 5' part, and also on the 3' part. And then this is extended during a DNA polymerase work. So by doing so, we can have the invading DNA, or a part of the invading DNA, in the CRISPR locus. And this is surrounded by these spacers, the same spacer bracket, because these are the same part that gets replicated here. So with this we can add more spacers on the DNA. Maybe this is for I don't know, another infection, previous infection, and this is also from this, and this is from another infection. So we are acquiring more possibilities of defense mechanism or possibilities. When we have this, then CRISPR can act in different ways. This is a lot of information and a lot of work, and I will go by I will not explain type 1 and type 3 more in detail because they are not that used, but they exist and they can have different effects if you want to use it on the lab, for example. But yes, for the type 2 is the more specific one, so it's the one more used. But all these defense mechanisms of CRISPR has these common parts. That is the first is the pre-CRISPR RNA is expressed. So all the repeaters and spacers are expressed in a single chain, like this here, here, and here. And then some RNase proteins bind specifically on the repeater parts, like this or like this. And they cut the RNA in a specific way. This depends on the type of CRISPR. So what we have is a mature CRISPR RNA. So we have the spacer, in this case here or here also in both parts, but cut it in the middle. And then, sorry, the spacer is the part from the outer DNA. And then the repeaters here, here, and here. And then using this RNA as a template for some DNases, in this case Cas9, sorry, they use this DNA as a template to recognize the foreign DNA and destroy it, so cut it in different parts. And this is the main defense mechanism. So they are acquiring, in the first infection, they are acquiring the knowledge of the sequence, the outside sequence, and then they are using it to check if the DNA that we have is the outside DNA, and if it is, they are destroying it. So this is the main idea of the CRISPR, and it's really similar to our immune system. So we are creating more antibodies, and these antibodies recognizes some antigens from the outside. So this is exactly the same, but they are recognizing DNA specifically, or RNA in this case also. As I said, the type 2 is one of the simplest ones because it only uses this Cas9 protein. That is a huge protein, I have to say, to do all the work, more or less. But also we have this trans CRISPR RNA part that is another recognizing agent in order to avoid missings here, or to avoid cutting our own DNA. So this is an extra recognizing step to be sure that we are not cutting our own DNA in this case. And I will explain this more. Okay, don't worry about a lot of things here. It's much easier because we already saw that part here from Cas9 to this CRISPR repeat spacer array. These are the Cas proteins necessary for the CRISPR-Cas9 mechanism, that is the type 2. And then we have also a trans CRISPR RNA. It's trans because it's the coding of this part, it's on the going to the other way around. So we have another production mechanism here that is not depending on this. So we have another leader here for the production of this RNA. Okay, the mechanism is exactly the same as I said. So the whole RNA of this part is produced with spacer, repeater, spacer, repeater. Then this is complementary with the tracer RNA that it's already produced earlier, because as I said, this is independent of this part. The production of this RNA is independent of this part. And while this is recognized by the trans CRISPR RNA, the RNase 3 that is from the cell cuts this part and gets stuck on Cas9. So this part of the trans CRISPR RNA, it's necessary for the Cas9 to work So this is an allosteric activator. Because Cas9 has some other subunits that are these two that are the main ones to cut the foreign DNA. But for this to work, we need to have this part, so the trans CRISPR RNA inside the Cas9. So while this is inside the Cas9 can recognize this other, the DNA that matches with the spacer RNA, and then this gets cut for the deactivation of this DNA. And this is again only happening if this trans CRISPR RNA confirms us, in this case, that this comes from the CRISPR array. If not, this will not happen. So this is an extra security mechanism, I will say. Is this clear? Because this is used in the laboratory for editing genomes. So I will talk about this part, and I hope it is clear enough here. Well, I hope the answer is yes, because you're not saying anything. Yeah. So well, I'm also putting a lot of references here. I hope I will give you this presentation at the end, so you can check. These are more specific for this part, the references that I'm including on the slides, but I also give you some more general papers in the mail, I think, but I will also give you this presentation. So I will explain you the genome editing tool in the example. So how we can use the CRISPR Cas9 mechanism for editing the E. coli, or E. coli, I think, because it's a Latin word, so I have to say E. coli, to make some resistant genes. Okay, so we have this E. coli, and we can transform the cells with the CRISPR loci. So we are adding this whole part here inside the cell. We already know how to do that because of the previous training in principle. So how to make competence and how to infect with a vector to the cell, and this can be produced, like if these proteins were our proteins, what we want to produce. And this gene, so a part of this gene is what we have as a spacer here. This is important because this is the main protein in the 13S ribosome protein, well, protein complex. And it's what gets attacked, let's say, by the streptomycin in this case. So this is what we want to change, this genetic area. And following the protocol, we said like this: the Cas9 cuts the gene that we want to edit, in this case this one, and then the cells starts repairing the DNA. Because the CRISPR cuts, or the Cas9 cuts both strains, so they cannot be, like in this first part here, they cannot be reconstructed again because it's cut completely. Okay, so we do not have this part to replicate, and the body does not, well, the repairing mechanism does not know if this has a missing part here, because it's not like in diagonal, the cut. So what the what the bacteria in this case does is finds an homology model in this case. So DNA that it's similar on the extremes, that they can be used as templates for the reconstructing this part that is cut. So what we can do is float completely the cell with our template DNA. So insert a new template DNA in the cell, and while the cell is trying to find the new homology, we give them what we want to be there. So the body insert this part, and in this case, if we want to do the resistant E. coli, what we give is this mutation that is not here because it's patented. And then these mutations makes the protein different enough for the streptomycin to not act. So now what we have is the gene completely modified. It's only one position, but we modified this exact position by the modification that we want to do, and it's not needed to have the restriction enzymes in the nearby area, or we don't need to, because this is really specific. We can say, okay, cut exactly here, because we know how CRISPR works, and we know where the Cas9 in this case will cut, so we can create the new genetic part that we want to include or to change doing this homologous recombination. So this is really good for cutting in some genetic space that has no no markers or no restriction enzyme areas in which we can cut. And this protocol is extracted from here. So we can buy in Amazon a kit for doing CRISPR if we want in the house. The only thing that we need extra about is a microwave and the fridge. Sorry, a freezer. So I found this and I was stunned for, I don't know, an hour. We can do CRISPR in our house for 170 euros. So, and I have to say that the protocol is really well explained. So, well, it's funny for me at least. Another example is, not always Cas9 is used. In this case, I found that the Cas13, it's a recent discovered Cas protein. This Cas protein is different than Cas9, does not need the trans CRISPR RNA, and is not specific in the cutting process. So what it does is, while the Cas13 is activated, it cuts everything around RNA, obviously. So if we add the Cas13 and the CRISPR RNA in a DNA sample, well, RNA sample, so we transform the DNA sample to RNA, and then we add the CRISPR RNA and the reporter, and one reporter RNA. So one thing that we can track if we have at the end, we can establish a protocol checking for if we have some DNA or RNA in the sample, because the Cas13 will cut everything, also the reporter. So if in the DNA sample we have the specific sequence that we give in the CRISPR RNA sample, we can check for this sequence. Now imagine that the DNA that we want to check is from SARS-CoV-2. so the coronavirus that we have now. If we have the coronavirus in the sample, we can check if there is a reporter later on or not. So we have a mechanism to detect SARS. So the COVID-19, and this is already done. I mean, they are doing it right now. The mechanism is not new. It was used for some other virus, and obviously this idea is not mine. But they are already doing this mechanism to detect the coronavirus in samples, in human samples. So this is really nice, I think. And this also can be done if we can do it, in the previous one we can do it in the house. These do not need the PCR machines. These do not need any of this, simply a well-engineered reporter RNA. In this case, it's a RNA with an antibodies. So we have the paper strips, like the older pregnancy checking strips that has 1 or 2 lines depending of it's positive or negative. So it's the same mechanism in this case added to the reporter RNA. So this can be done on the fly. It's like 5 or 10 minutes. But obviously this is not the already established. So, well, I think it's a really nice use of the CRISPR capabilities in this case. I think this is the whole part for the CRISPR. I don't know if everything is clear. I didn't explain far the CRISPR part because it's extensive and we have a lot of mechanisms, a lot of new things happening right now in the CRISPR area, and a lot of different Cas that proteins that can act differently. So new mechanisms can be established, and new things can be happening in this field like every day. So you can read about new CRISPR capabilities in the Nature Communications for sure. In the next one, for sure. I mean, there is always some articles about these new CRISPR uses or capabilities. So I explained to you the main parts and why this is working really well for some things. And then if you want to know more, I think you have to read more, because this may be this can be really specific for one protein, one Cas protein, and a specific one type of CRISPR mechanism that is not the 2, is the 4th that I read that is a new one, that is the 4th, and is not included in the review that I used for the training. So yeah, there is a lot of things going on the CRISPR part. So I really encourage you to do your own research and to read about this, because it's really extensive and you can do a lot of things. Also using CRISPR interference for the induction of some genes by using an inductor, or so adding murine to the sample or to the bacteria. This will act as a precursor using a CRISPR mechanisms that is much better in some cases than some induction mechanisms using IPTG or some other used mechanisms. So I really cannot explain to you all the uses, because I cannot understand all the uses, because this is really a going on field. So what do you think? We can do a break right now. We are going on like 45 minutes more or less. So it's okay for now to stop. I think that for 15 or 20 minutes we will be finished, if you don't ask much. So what do you think? I don't know if I can hear you. I think so, but I cannot.
Yeah, we can hear you.
Okay, okay.
There was some delay between Discord sound and the YouTube video, so I just moved
Yeah,
to
the YouTube video is
YouTube.
not on sync, but it's okay on the Discord live.
Yeah, the Discord is okay.
I haven't tried it. I can check it. Yeah.
Okay, okay.
Yeah, it looks fine.
It's okay, yeah.
Sure, and you get more or less everything, or not more or less, I mean, you get everything, if not, ask.
Well, do you hear me?
Yeah, I heard you, Lorenzo.
Okay. Well, because I have a question regarding the initial part.
Sure,
I don't know
yes.
if I can ask now
Sure,
or
yes.
later.
Go ahead.
Well, regarding this lytic or lysogenic cycle,
Yep.
is it related to what we call normally the incubation period?
No.
Yeah. Let's say referred to the whole cycle, or just, for instance, within the lytic or within the lysogenic, what it was called latency in the cycle.
Exactly. This is not related with the incubation cycle because while you are in the, if you have a lysogenic infection, in this prophage infection, I mean, this is the typical example of the HIV. So this can take like years of developing, but this latency period is normally related to this part here. So in the lytic cycle, also in the lysogenic, because it's an extension, is the time that the virus need to produce a lot of virions and break the cells. So while you are infected, but you don't have too much synthesis of the virus, in this phase here, you are okay, but you are already infected.
Yeah.
You only have the symptoms here in the lysis, while your cells are breaking. So this depends on the virus. This can be faster or slower, and this is this latency step.
Okay. Yeah.
So
I have a more simple question actually.
Sure, go ahead.
Which is I've always heard about CRISPR as a gene editing tool,
yes.
But so this already existed in bacteria, and humans just harnessed it to do whatever they wanted.
Yeah, well, the CRISPR was well. I think I can hear myself in your in your. Yes, thanks. I think it's Lorenzo's one. So.
I'm muting.
No, sorry, it's my fault because I checked the YouTube thing. Sorry. Okay, perfect. So the question was about the CRISPR. CRISPR was a mechanism, a defense mechanism from bacteria and archaea. The humans, let's say, or the mammals, the organisms that has more cells, the pluricellular organisms, has not this mechanism. So we use like the PCR mechanism. So we use this in the lab, and we know that this is from bacteria. So we add these genes to the bacteria in the lab. Maybe depending on the bacteria, this is already there, but we want to add the spacers and the Cas genes specific for our mechanism that we want to do. I don't know if I'm asking properly, so I'm answering properly.
So in fact, we are just taking advantage of a mechanism that already exists in the bacteria, and we just use it to our favor.
Yeah, exactly. But if this mechanism, I don't know, if we are using in the lab a bacteria that has not this mechanism, we can add it to the genome. So adding this to the genome, we are adding the mechanism to the bacteria. So we don't need to use a specific bacteria for doing so.
But and is there any difference on using a bacteria that already has it, or?
I think it's more specific on what you want to do than for the CRISPR. Yes, there is some differences. As far as I know, CRISPR is in theory really nice, but in the practical part, it's already being set. So maybe for some bacteria, while you add this, the bacteria detects this as a foreign or something, and it's not do not get included on the genome. So it's not that straightforward as I'm saying it, maybe. But I think that the bacteria that you're using on the lab normally does not depend on the CRISPR mechanism that you want to do. So maybe it depends on the conditions that you want to replicate this. Maybe if you need a bacteria that resists higher temperatures for the incubation or what you are doing, but it's not as depending on the CRISPR that you want to use later on. I hope I answered more or less okay.
Thanks.
Okay, so if there is no questions, I will give you, I think, 5 minutes, because yeah, I think there is no much things to. I mean, I think in 20 minutes or so we can finish this part. So I will use these 5 minutes to upload the presentations for you to have, and then later on, after these 5 minutes, I will ask you again if you have any questions on regarding the presentation or this. Okay.
I'm muting.
Ah, no, sorry, it's my fault because I. Well, welcome back again. I will wait, I think, one more minute if anyone is still on the toilet or something. And well, this next part will be, I think, easier to understand for everybody and shorter, I hope. Yeah, no, for sure it will be shorter. But I think it will be the most interesting part for a lot of us. So if you check the schedule, now it comes the screening part, so the Phage Display part. So as you know, in 2018, this was the Nobel Prizes in chemistry. We all know Frances Arnold work, so I think this is already really known, the directed evolution protocol. But there is one part in the directed evolution protocol that we do not know much, because in the previous biology trainings we discussed about cloning, also producing and purifying proteins. That is an important part, maybe not the purification in the directed evolution, but it's important to test the proteins. We know how to produce a lot of random mutations using error-prone PCR. So, well, that's already done. We can produce lots and lots of new mutations, like millions of new mutations in one step and in one pot. But the problem is that we cannot test properly all these mutations. So we need an efficient screening method, and the first one was developed by George Smith, and it was the Phage Display. It was firstly developed for the testing of the new antibodies. So maybe it's not the best fitting screening method, but it was the first, and it was really a really new technique. So it's what I will present you today. And obviously, because this is the training biology, I'm also explaining this to you because we use virus machinery for doing the work in the Phage Display screening method. So first of all, why we need a high throughput screening method? Well, that's already said, because if we can test the proteins in 96 wheel plates, for example, but if we are producing thousands of mutations in each round, directed evolution round, maybe 96 is not a huge number, I will say. So we need a test or a method in which we can test thousands of variants. And finally, if we are in the directed evolution scheme, we have some variants here, so the mutants done by, I don't know, error-prone PCR. And then we want to screen these and select the ones that are good. But then if we want to do a second round, we need the sequence of the protein. What mutations are included here? So we can test, yeah, the protein 12 here in the plate. It's the one that is good, but then we need if we want to use this as a new template, we need to know which mutations are here, or which is the DNA or the protein sequence. Obviously, to sequence sequence a protein is not easy, and we cannot do, I don't know, mass spectrometry for each one. So this is not an option. We need a method that links the protein and the DNA together in order to select the DNA that will be the in use for the next round of directed evolution. So we need a method that is good for testing a lot of variants and also that links the protein with the DNA because it's easier to sequence DNA. So the main idea is, okay, we have a batch of mutations here by error-prone PCR, and we have a winning protein, that is this. But we need to link, in some manner, the protein with the DNA, with the mutations. And if possible, also, we need to code the DNA because it can be degraded really easily. So, because if we want to do this fast, we will produce the protein, destroy the cell, and then test in the whole cell proteins. And a lot of these proteins are DNases, so we need to protect this DNA. And also with the protein attached. So this is a really not easy thing to do. But we have something that does this automatically. And we need to be automatic, because if not, we will work for years and years for one round of directed evolution. And it's the virus. The filamental virus that I showed you in the first or the second slide, it's only 10 proteins inside the DNA. And while if this is produced by the cell, this will produce some particles with the DNA inside, well protected, and with some proteins outside, coding the virus. And if we change these proteins outside, we can attach our new protein, the one that we are testing. So if we change this G3 protein, that I will go faster to the first. Well, I think I have the next slide, I think. Yes. So if we change this G3 protein and we add here the, well, I think it's this one here, but imagine that here in the P3, the protein 3, that is the one that will code our phage. We add here as a fusion protein our protein. The particle that we will have at the end will be the virus protecting the DNA and with our protein outside. So the protein and the DNA, it's linked, and the DNA, it's protected, and this will happen automatically if we add all the other proteins that are necessary for the assembly and the replication of the DNA. Also, because we are changing this P3 protein, the infectious, that are some proteins that are necessary for the infection, we will produce these new particles, but this particle will not affect other cells because this particle has lost the infection capabilities. So this is perfect for what we want to do. I hope this is really clear. I think it is. So this is what is done in the phage display mechanism. We change the protein, the code protein 3 in the phage. In this case, the first one was the filamentous phage, the P3, I think, like the protein in this case. And this is displayed on the phage. We can test this, and by recovering the protein, we are recovering also the DNA. And the screening method in this case, because it was mainly for antibodies, is like this. So we have a selector immobilized. This can be in a, I don't know, a petri dish. We add all the virions. The virions are created randomly. So we have generated the genes doing error-prone PCR, and we are attaching what we have, the output of the error-prone PCR, using restriction enzymes. Now that we know we can use CRISPR, for example, but not on 1989, I think. We attach here using restriction enzyme and ligase the genes that we come from the error-prone PCR. So every particle, every cell that is producing, bacterial cell that is producing the viral particles makes different proteins, okay, because it's mixed on the error-prone PCR. So each cell will produce different variants, and this is on the same pot. So what we have at the end is a mix of virions with different proteins and different DNAs. It can be thousands, depending on the procedure, but it will be probably a lot. And then with this selector immobilized, we add the virions. We incubate this some time, and then we release the media. So we extract the media, and the ones that are strongly attached to the selector are the ones that are working better. We can increase this stringency by incubate less time, for example, or in worse conditions, or by reducing the number of immobilizing selectors. So with this, we can select more and more efficient proteins to recognize the selector. So we are selecting better proteins and also selecting better DNAs for the synthesis of these proteins. Is this clear? Because this is the main mechanism. That's all. So obviously this mechanism has some limitations. It was the first one, and it was really, really good, but now we have much better mechanisms. But the idea is more or less the same. And the main problem that we have here is that we cannot include whole proteins normally, because bigger proteins disrupt a lot the proteins on the code and makes the virions to not properly be created. So we do not have the virions if we have a big protein. And big protein, I will say like 100 residues. So we only can use some parts of the protein. So this is not the best thing. Also, not all the proteins will work here. If we have an enzyme that needs to be opened and then the active site is inside the protein, this will not see the selector by any means. So we cannot use this method to select, I don't know, MAO-N, for example, because the active site is buried inside the protein. So, and it's also a huge protein. So nope, we cannot use that. And also some proteins cannot undergo the process of being translocated into the membrane, because I didn't explain it, but these proteins, while generated, they need to be outside the bacterial membrane. And then these, while the virion is going outside, these are recruited. So this process going through the lipid layer is not friendly for some proteins. So we need to make sure that these proteins can survive this. Some other problems are, for example, that if we have proteins, human proteins, for example, these proteins may have some lipids, some sugars attached, or some modifications attached, and the bacteria cannot do this. So we need to do so with the yeast. The yeast can introduce the modifications in the proteins like we do. like the glycoproteins, for example. But maybe yeast add so much. Also, that's another problem of this modification. So this is also a big problem. And also yeast are bigger than phage, obviously. So the number of organisms that we can test are not that much. One of the best screening methods are the ribosome display, in which we can use human cells, for example, and use the ribosomes in some way in which the RNA, while the protein is produced, the RNA at the end, it gets stuck. So we add some non-canonical amino acids, for example, so not amino acids, bases, that the ribosome cannot transcript. So the protein and the RNA get stuck in the ribosome, and then we can display, so test for these particles, and are the proteins and the RNA that can be translated to DNA in the same particle. So this is also a nice way, and it can be done in human cells, for example. So this will solve us the glycoproteins problem, and also the size problem of the protein. Well, maybe not too much, because if the protein needs to be really flexible, if it's attached to the ribosome, that is a huge protein, protein and RNA complex, maybe this is not the final solution, I will say. In principle, what the best solutions that we have now are the high-throughput droplet microfluidics, normally using lipids droplets, in which all the reaction and the DNA, so everything is inside the droplet, and then we can test the droplet one by one in the chambers with extremely precision machinery. So this is a really expensive method, but it's the best. I mean, you can add whatever you want. The protein is free on the media. The DNA is also free in the media, but because we control what it's inside the droplet, we know that the DNA will not be degraded. So this is the best way for sure, but it's really expensive, and not much laboratories can do so. But it's the principle is the same. We have the DNA and all the mechanism for the production of the proteins inside a lipid drop. The protein is produced, and there is also inside the substrate and some mechanism to display the activity, like some color change reaction or pH change reaction. No, I think in color, and then this is tested with normally lasers, and it's completely automatic. So it's like the NASA engineering, but it works really well. But it's really expensive, and you have to work a lot for designing these experiments. So I think it's everything set. It was like 15 minutes. I think this gets it's understood. I think it's a really easy method. Also, what I really want you to retain is that how nice is the idea to attach the protein and the gene in a virion particle, because I think that the idea is awesome. It's a novel idea. But and then the screening method is, maybe you already know that this is how this is done. But yeah, it's the screening method that these particles can go through. Also, because of the virion particles, if you want to add more stringency, you can add trypsin to the media. The Capsid proteins are really, really strong and resist a lot the trypsination process. So you can attack with the trypsin this link here by attacking the proteins and also the acids that you need to add, but you will not destroy the code for sure, because the virus, these capsids are really, really strong. So there is another way to add astringency to the selection method. So I hope this is understood, and it was all. I mean, I was expecting maybe to talk more, but I think it's everything well understood. So you are really nice, really nice students, I will say. So there is any question? I saw Curado typing, but I don't know if it's typing. Okay, nice talk, Miguel. Thank you very much. Just a curiosity for the method. Is there a known range of residues for which is this method working? Is there a known range of residues? You mean the number of residues that you can use, so you can display? Yeah, for sure it's known, but I don't know it. So use the Bible that I'm providing. For sure you will maybe hear in these reviews. For sure you will know this information, you will read it, but I don't know it. I know that because this was done firstly for the antibody, obviously this and a full antibody cannot be displayed here, and I think the full only one branch of the antibody also cannot be displayed here, so it's the half of the one branch of the antibody that is the extreme part, the part that it's detecting, let's say, the antigen. So I don't know, maybe it's I don't know exactly. No, for sure for us it's not applicable for sure, I think, but it's really nice to understand how this works, because all the other mechanisms are using this as reference. So if you understand this mechanism, you will understand how the ribosome display works. That it will be really easy to understand if you read it now, I think. And it's like I said, like changing the RNA in order to get stuck in the ribosome and then use the same this screening method. So also the machines that are working now, so the typical machines that do directed evolution in the lab that are really, really expensive, use this method. So the the wheel plates, but because it's a machine, they can test a lot of things at the same time and not being tired, let's say. And also they can know they can have on memory which test corresponds to which colony on the Petri dish. So it's easier for them, for the machine, to link the experiment output from the input Petri dish. It's also done, I mean, on the lab. This method is still one of them, or maybe the most used for directed evolution, but not high throughput or higher number of variants, but it's still really used and you need to make sure that you know which colony, bacterial colony, corresponds to which output. So normally you take a photo and then you add this number 1, 2, 3, 4, 5 in the photo, and then you mark the petri dish in order to know which one is and corresponding to the photo. So this is still a method really used because it's cheap and it's using things on the lab that we have. But if you want to do a directed evolution for using, I don't know, 1,000 or millions of variants, you need to use a screening method or a machine that does the work for you. But it's, in both cases, it's really expensive. But the screening method, maybe it's not that much. Okay. I see there is more people typing, so I will wait a bit. Okay, Silvia. Thanks, really nice tutorial. Thank you. Okay. The droplet-based screening method was the one used to generate the most evolved retroaldolase variant, 8F, which was catalytic activity is close to natural aldolases, just to mention. Yeah, it's in that, if I remember well, on the I don't know, it was the Zurich University. They are also developing some microfluidic systems, and yeah, it's already going on. It's the one with the best results because they can test a lot of variants without the limitations of this screening method like Phage Display. So it's a really cool method, but it's still really complicated. So thank you very much for being here and to, I don't know, get some interest in this biology part. I know it's a strange part maybe. Now you know some more about viruses, how these are divided in families is not as straightforward, I will say, like in some other organisms, because they are really strange and really dependent to the host. Thank you very much for being here, and well, I don't know. Remember, if you are really interested, you have the CRISPR kit on Amazon. I don't know. maybe it's fun. So, well, thank you very much for being here and for all the comments. And I don't know, see you soon or not, I don't know. Thank you very much for the comments. Well, I will shut down the streaming there.